So the first issue is how can we predict, and I'm going to skip through a couple of slides simply due to the fact that I couldn't get everything done in time. So bear with me, I'm sorry for that. So life is diverse. What is common to life are cells, genes, proteins. We talked about that. Those are just the parts of some common principle, and the common principle underneath is evolution. Life is evolution. Evolution selects diversity. Evolution selected what we see as life. Who invented evolution? Well, you know, it's clear who did that. But if that is so clear to everybody, what's the Wallace-Darwin theory then? And that gets into, well, you may all know Charles Darwin published this book, The Origin of Species, in 1859. That's the front page. That is the only figure in the book. So it's a heavy volume. It's not easy. It's not an easy read. And this is the only figure, right, that he has, and that is the origin of species. That's the tree of life. That's the idea. Now, this idea brings us back to similar sequences have similar structures. So I said number of residues, percentage sequence identity. If you have 100 residues aligned, 33 of those identical in the blue, the two proteins essentially have a similar 3D structure, right? When we look at single changes, let's just look at a particular protein here, an oncogene KRAS. That's the way it looks. The coloring is showing you the orientation of this molecule. And what is overlaid, and that's a slide from Andrea Schafferhans. Now, on top of that, now you actually see two structures superposed here, and they differ by one particular residue, and that is at the position 12. There's a G and a wild type, and that's varied to a C. Okay. Now, do you see a difference? So, some differences are here. For instance, that this strand here is starts a little bit earlier or ends a little bit earlier, starts a little bit earlier here, later here. But you do see that in this particular case here, the binding site looks very similar. Now this looks very similar. In this particular case, leads to a different function. This causes cancer, this variant. So that is exactly the type of bad variant that makes a big difference. You see on the overall structure, it is not a big difference, not even on the level of the binding site, okay? Now, let's go to another protein here. RASH in Lyme is a protein that has 85% sequence identity. Again, you essentially see the same. Now you see that the binding site actually looks rather more different. We get further down to, so far Lyme was still, RASH was still in human. Now this is a fly protein here, 28%. You do still see that a lot of these strands are at the same places. The helices are at the same places. Again, there is a slight variation, so these helices look slightly different. The binding site now begins to look very different, but there's overall, essentially, they are very, very similar. And even if you go further down, in purple here, we go into the bacterial world, at 90% sequence identity to the original one, you still see that essentially, so these fingers here look similar. Even the helices now, here you see that in the purple the helix really has a different winding. So it's slightly, the connection of the helix is done differently here. But essentially the objects are still there, right? And essentially not only that, you also still see a binding site. The binding site binds something else. The binding site looks different, but it's the binding site at the same place. So there's a high conservation. And this essentially is what is conserved. There's a motif, or motif may be the wrong word. You have a basic way in which you make it work. So you have all of that as a buffer or as a skeleton for the binding site that sits here. And you maintain that through evolution because it works, okay? And then you put slightly different substrates here, so slightly different binding sites, you slightly change the details, and that is good enough to sort of use the same thing and do something else in a different organism, okay? And why is it good enough? Essentially because the pressure is to reuse it and do something somehow similar. So genetic information is packaged in chromosomes. Here the 22 human chromosome, or 23 human chromosome, including the X. Now changes happen in all kinds of ways. So one way of change would be you insert some stretch of sequence into one of these chromosomes. You could do the opposite of insertion. You delete a stretch. This is a duplication. You simply, when you copying the genome code, you copy a part of it and you double it. So you essentially take the copy and just cut and paste it twice. So you hit the paste button twice on the computer, and the machinery does essentially a similar mistake here. Inversion means now there is a direction in which the genome code runs, right? And if you sort of cut and copy and put it the other way around, there's an inversion here. So then there are translocations. Translocations essentially means you take it out from here and put it in somewhere else, in some other place in the chromosome. And the pieces that you can take out have all kinds of length. So these pieces that are taken out could be genes. There could be fraction of genes, and ultimately it could be a single residue, right? And the part that we talk about today is really SNF, single nuclear variant. So that is in the difference between these two. You see that one letter, so this is the double-stranded helix here, double helix, the genetic code. The only thing that differs between the two is this G. Here's an A. And that's a SNF, that's a single nucleotide variant. Historically it was called single nuclear polymorphism. Let's not get into what it is. Essentially what it is one nucleotide is different. Okay? Other than that, these two are completely identical. They change by one letter. It's a word with one letter difference. And again, you know that this shows you that you can change 67 residues in 100 and you don't change structure. I showed you examples that a single change does not really matter in some cases, right? Now, the idea somehow of mapping genotype to phenotype means you map your genome, that's your genotype, that's the way your genome sequence looks, to the phenotype. This is the way you look, or the way you appear, your traits that are not the inherited traits, that is not part of your upbringing, the way you looked into life. And somehow what we're trying to see, since we know the human genome now, from the changes between us, what can we learn about how we differ? Okay? That's one of the stories that we are after. And the first step for that ultimately is, can I understand for a particular change, for a single residue change, for a single letter change, whether it has an impact on function? Yes or no? Can I predict that, right? By the way, these SNPs, or these SNFs, these single changes, and studying sequences, ancient sequences, got us some rather amazing realities. For instance, so this is not our group found out, Science found out, that's a group in Denmark here, found out that there was an invasion into the North Americas long before the time that we believe happened, that created the Native Americans and Inuit. So it was 5,500 years before we believed that what then became the Native Americans moved there. And that was the only way that was found is essentially through some genetic evidence that was found in Greenland in this particular case. Here, a remarkable study showed a few years ago, just sequencing African genomes, that in fact when you look at African genomes, when you look at genomes in the group, what you might perceive is that we in this room, we all differ. And you may sort of feel that whenever you are from some country, so we happen to sit in Germany, so when you look at a bunch of Germans, that they are sort of more similar to each other than they are to a bunch of Italians. So yes, Germans differ, yes, Italians differ, but somehow when you look at all the Italians and all the Germans, you find something in which they are sort of similar to each other. And then, maybe you go to the Swedes and then you see more differences between Italians and Swedes maybe. And then you go to Asian countries and you maybe see more differences even, right? And then when you go into Africa, so we believe that Many people believe that life started somewhere in Africa, or the Homo sapiens started in Africa, and it started in the area that is currently populated by Bushmen, or sort of the original native population, not far away from South Africa. Are in fact Bushmen, and I don't know whether you have seen Bushmen, but Bushmen, for me, they look very different from me. So that's the way I look at Bushmen. And they have languages that I cannot repeat. So they have these click sounds. You cannot even read. I cannot even read their language. Have you ever heard these languages? Can anybody? I don't know whether you know Miriam Makeba, or so when Miriam Makeba sings her songs about click sounds, so I will not even try to do this. So there are people, friends of mine, sort of they try and they dare to. It's remarkable what you can do with your tongue, right? But so they look different and they communicate different. They're just, it's a different. And they somehow, from where I'm standing, they look all alike. And the reality is that genetically the opposite is true. These Bushmen differ more from each other than the Chinese and the German, okay? Genetically. That's the remarkable finding. So for all the differences we see between us, the real differences happened a long time ago, because, again, people argue this shows you that life started there. The full diversity was there, and essentially, why is that the case? Ultimately, I consider it a surprise, and I keep being surprised by this finding, but the rationalization, again, of this surprising finding is when you have a group, then you have so when Homo sapiens started, there was a certain diversity in the group, in the original Homo sapiens group. Any group that goes out from that has a lower variation in it than the original group, because it's a subset of people that goes out, and somehow the subset clearly has a lower variation. Why then over many generations things don't change? Ultimately the answer to that is because humans do not have that many generations yet. So if we waited another million years, then we would change, maybe turn this around. But currently that seems to be the reality. And again, this is a finding that happened only through looking at SNFs. At these single residue changes, that's the way they trace groups of people, and that is the way they trace these differences without saying that this particular SNF, this particular letter change has an impact. Just saying, I can follow that SNF and I see it belongs to that population, and that SNF to that population, and then I can study how much diversity is within a population. That's ultimately what they do. And here's a work that is mostly done by Sven de Pabo, where they defined a new species, the so-called Denisovin. So he discovered Anatoly Derevyanko discovered in a cave in Siberia an organic, something that they thought actually was some wild animal. It turned out to be human, a part of a human, a little bit of genetic material, enough to come to the first time in history that a species is defined by sequencing. So they found out that this species had not been described before. It's prior to Neanderthal, is a species that Neanderthal and Homo sapiens have in common. And again, so there is the species of the Denisovin, according to this Denisovin cave, and nobody has ever seen it. So all the genetic material that was there was done to sequence. There's nothing left of Denisovins. Nothing that we know is left of Denisovins. Actually, they found something later. So let me get back into the type of changes we have. Again, in this first example here, we have this one single variant in a non-coding region. Coding, non-coding, essentially coding part that gets to become the protein. Non-coding essentially is not becoming a protein, right? So this is only on the information level. This becomes a protein. So this first variant here on the level of the protein would not be visible. So between the original wild type and the variant, the protein looks the same. Okay, now in the variant 2 here, we have a variant in the coding region. But this variant here is such, it changes the G to an A. Remember, that's always three nucleotides that make one amino acid. Okay? And it turns out, since you have 4 different amino acids, so essentially you have 4 by the 3, so you have 64 possibilities. You have only 20 amino acids, because some of these triplets are used for, several triplets are used for doing the same amino acid, okay? So you have some redundancy built in. And what you see in this particular case, the case is chosen such that the amino acid GAA is in fact the same as GAG. That means that you have a sequence variant here in a coding region, but it does not affect the protein because it's the same residue that is done, because of the degeneracy of the redundancy of the genetic code. In the third example here, I have one, the GAA and GAG are the same, but AAG is different. So this variant here now changes the amino acid sequence. So now I have a change. The variant 3 in fact makes a K here, while all the others are on the level of the protein, not visible. Okay? So for the remainder part of the lecture, I'm going to talk about these here. So the changes that actually change the protein sequence. Today they are typically referred to as SAVs, single amino acid variant. These words are not important. They can affect function, okay? Here are some examples. They can lead to diseases. That is the first one. That was sickle cell disease, where a single change of a E at position 6 to a V in fact leads to sickle cell anemia. It's a bad disease. Now, we could try to predict whether a particular variant leads to an effect or not. And at this point, there are over 150 distinct prediction methods out there. In the time that remains, I'm going to briefly give you a few ideas of what they focus on, and that's where we can get. And then I'm going to give you an overview. By the way, how similar are mice and men? And I'm just quoting a book here. That's why I call it men. Any answer to that? So men are furry, mice are furry. Mice don't know what to do, men don't know. Any idea about numbers? Anybody? Venture?
90%? I don't know.
Yeah, so this is what you very often hear are numbers when you compare letter by letter the genes. You have some numbers that range in this ballpark. So 90% essentially is exactly the ballpark, right? Now, this is, however, the genome view. And on this genome view, mind you, only 1% of the genome is actually leading to proteins, right? So on that level, this number is sort of roughly true. But if I get to the level of protein, the story is actually slightly different. So 70% of all the proteins are more than, so this is the 70-70-70 rule. And there's a 170 missing. So how do I establish that two proteins are similar? So first, my first threshold is to say I have protein A and B, so I have to first establish how much do they have to overlap minimally, because we have this domain issue, right? And we have sort of what I'm looking at is I say that two proteins are similar if they overlap for at least 70% of their length, of the length of the shortest. Okay, then I call them shortest or longest? No, it must be the longest. So if two proteins at least match to 70% of the longest, then I call them they match. Okay. Now I ask, do they at least have 70% sequence identity? And then I ask, how many of the proteins actually have that? So that's the 70-70-70 between mouse and man. So 70% of the proteins have more than 70% sequence identity over about 70% of the longest when I compare them. Okay? Now, interestingly, by the way, what this one shows is that the same is true for rat. Mouse and rat is a very, very similar number. And surprisingly, for me at least, I was totally surprised. So human, I believe, I can't remember whether it was more or less, I cannot remember. Mouse, so it's the 70-70-70 thing. And I'm just quoting it here the same way. Because I cannot quite remember. It was a very similar number. The surprise for me is that was also 70, 70, 70. Just a little bit less, or a little bit more. So the mouse and rat on this criterion were just a little bit more similar to each other than either of them is to human. That for me was a big surprise. Anyway, so how would you develop a method that predicts the effects? Any idea? Before you say machine learning, any biological idea what you would look at? Have you taken away anything from the biology? Any signal, biological signal that you would look at? So you're asking, again, so we have a lot of sequence variation that does not matter. How can I distinguish between the one that matters and not? And the first method actually is called SIFT. And since the translation of SIFT does not give anything away, I can put this slide up. But SIFT is the biological idea somehow that I'm looking for. What would you look at? So, yeah?
Whether I'm in the coding or non-coding region first.
Yes, so here we are discussing only SAF, so we are only discussing things that are in a coding region and that change the amino acid. So those are the changes that are essentially shown here, right? And here is shown as identity, but 1 minus identity is change. And again, you see that you can change a lot. So the entire blue region is the change that is possible without really affecting structure. So how can you, for one particular residue at one particular position, predict whether it has an effect or not? What would be the simplest biological signal you would look at? And anybody who says machine learning, that's fine. We're going to get to that in a minute. But what's the simplest thing you could look at?
Well, yeah? With evolution, if you have other proteins with different SNPs in that position, then apparently it doesn't.
And that's exactly the idea of the first method that was developed by Pauline and Stephen Hanikoff, that looks at an alignment, and says, well, so there are these blue regions here where it's entirely conserved at that position, and if you change a residue that is entirely conserved, you do damage. If you have the change at a position where in the family of related things that you believe do the same thing, you see many, many, many different residues, then this will not have an effect. You simply look at conservation, okay? And then sort of the next step of complication is you look at change or probability of conservation according to if that, for instance, is a hydrophobic residue, and you replace a hydrophobic residue, somehow you know that the biophysics of hydrophobicity, hydrophobic to hydrophobic, is somehow similar. So then you sort of look at biophysical features, and there's a matrix underneath, Blossom matrix, that says when proteins can exchange at particular positions, which residues they can exchange without doing any damage, typically, or not without damage. What you observe in nature, right? So the type of changes that relate to what we tend to argue are the biophysical features. If it is one of those, you say okay. If not, you say it's not. And that ultimately is the entire idea. In PolyPhen, the same idea is done, but the way the math is compiled here, and this is from Sunyaev, in the way the math is done, that is different. So when I say whether you're conserved at a position, I said there are these blue things that are entirely conserved. So the PolyPhen brings a good math about what is expected by chance. Looking at the entire similarity of the entire, you imagine this family alignment to be a matrix, and then you compile at every single position what's the expectation for a particular value at that point. And that expectation is something that you can simply compute mathematically, okay? And that is sort of what PolyPhen does. SNP-S3D essentially uses a signal like that, but then says, well, whether you're outside or inside of the protein, what is in effect differs, okay? And when I have a three-dimensional structure of a protein, I know whether I'm on the outside or not. I know what kind of bonds I mess up with a new residue, and that is trying to measure. SNP-S3D tries to measure that. And with this, we essentially threw all the early methods that existed. These early methods have very, very few parameters to fit, and therefore they don't need a lot of data. They essentially can make a prediction without doing much, so without fitting anything, right? And that was in fact historically how it started, because there was not a lot of data there. Now, when you measure the effect, the problem is you have effect measured on very different levels. So one level where you really see that these two mice look different. And in this particular case, there is a single letter change that leads to the difference in these two mice. Okay? In some cases, you have measurements of some enthalpy of a delta G of binding of two proteins. Okay? This is a very detailed biochemical assay, a biochemical measurement. And then there are all kinds of things in between. Now, when you look at effect variance, you have to sort of find a way to integrate the data on all of these levels. And that comes along with a lot of thresholds and with a lot of additional definitions and with a lot of work. And in fact, Jana Bromberg here, who developed the first machine learning method in this field, most of the time she worked on creating those data, to create a large data set that you could actually use to start machine learning. Because just a bunch of is not good enough for machine learning that existed. And then after several years of work, ultimately she aggregated a data set that mostly was done by this database here, was one where you had about 40,000 single amino acid variants. So one letter change for 40,614 or 40,000. You see the effect of that. There is an effect. For about 14,000 other variants, of course, you see there's an experimental result that they are neutral, meaning they don't do anything. Okay, so visually put here, most is red, most has an effect. Why do you believe that's the case? Why is it so imbalanced? Is that because most things have an effect? Yeah?
It's probably easier to show in an experiment that something has an effect and it remains neutral.
Absolutely. It's easier to show and it's much easier to dare to publish. So when you say, I have measured that effect, it's a very clear-cut definition. Yes, maybe you made a mistake. Okay, but you provide the conditions under which you do it, and you repeat this experiment several times, and then at some point you believe yourself. You have measured this under these conditions. Maybe under some other conditions it wouldn't happen, but under those conditions I've seen it, I've repeated it, I know it. If you say this has no effect, then you always have to say, well, no effect under the conditions that I measured. But no effect will get into the literature, get to machine learners who will call it neutral. But it may not be neutral, so it may be neutral for binding this protein, but maybe down here something changes. So the variant here may affect another binding, right? So publishing that this variant down here is neutral would not be right simply because I did not try all possible conditions of this binding. So one thing is more complicated, another thing is more daring to say something is neutral. Because you always have to go to the small print, and we all are not very good at reading small print because we overload it, right? Anyway, so you get a very unbalanced data set. And how does machine learning actually handle this unbalanced data set? We have had many examples of unbalanced data sets. Many data sets are unbalanced. How can you balance it? If you had a data set like that in machine learning, how would you balance it? How could you balance it? There are many, many ways of doing it. Just come up with a couple of ones. I showed you one. We spoke about one. Just name some others. Have some other wild ideas. So what you want to do is you have this green part here and the red part, and you essentially want both pies to be equal sized. Yeah?
Well, you can do. Well, either you can just use the same amount of proteins from the ones that have effect. That's the one.
So you sample. So that's statement number one. Let somebody else do the or. So the either. This is one. So you simply randomly choose from the red the subset that is this size, right? So meaning that in this particular case here, let's just call that a quarter, the 26. So meaning that half of the data you will throw away, right? Yeah, what else could you do?
Or I could do the opposite, oversampling the green ones.
Exactly, you could sort of have duplicates of the green ones. In this particular case, you would roughly triple it. So you would have you would add 2 duplicates of the green one and throw that at machine learning so that at every single time step, it would get an equal amount of red and green. What else could you do? Oh, there was an or, okay.
If you're using an optimization algorithm, for example, a nonlinear optimization algorithm, you can, I think there exist schemes such that you can do weighting, which means that...
You said that the other day, no? No? Yes, so you could weight.
So the gradients that you compute for the minor class gets a higher weight and difference.
Exactly. Exactly, and this is in fact different from different sampling. Or it could lead to a similar result, but technically it's different. In terms of the equation, it's different. It could really lead to a different point, and very often it actually does lead to a different point. Any other idea? Yeah?
We can create synthetic data, but I'm not sure how.
Yes, we can do that. So let's just consider this example. And most of it is on the slide. I should have started possibly slower. Let's just assume that you had 2 proteins here. And for both these proteins, you have experimentally measured that they are enzymes and that they have a particular enzymatic activity. And that you actually find experimentally for this one and for that one that they both have the same enzymatic activity. Okay? So then you may argue, well, wherever they are different, I say those residues do not matter. Because they do the same thing. Okay? As long as these sequences are similar enough, I can argue that wherever they vary doesn't matter because they do the same thing, right? It's not quite simulated, but something that is not annotated either. In that sense, it's something created in the computer, or by let's call it differently. It's something created, it's a data set created by computational biologists. And in some sense, you could argue this is as close as you can come in this world to simulated data that is helpful. So this is an example. And in this particular example, so when we merge the data set, you see that we actually, with that, we can create a data set that is now roughly balanced. So we have a similar set of letter changes that have an effect and those that are neutral. And overall, this is sort of the ballpark of accuracy that we get. And there's sort of the second generation of methods that use these sort of larger data sets and begin to really use machine learning devices. Those are 3 examples for that. And I'm only going to talk about SNAP today, and I'm also not going to talk about the 3rd generation. Instead, I'm going to dash for an application of SNAP, to discuss an application of SNAP. And SNAP 2 comes from Jana Bromberg. So the first decision was, we tried a variety of machine learning packages and ended up with a neural network. And in this neural network we have the maybe not so surprising architecture. The image has been used for other things, where 1 unit codes for effect and 1 unit codes for neutral. The input here is a consecutive set of residues, shown 13. And for each of these, you have 21 input units to have a spacer. This is actually not quite right, because in this particular case, the central has 22. And it has 22 because you have the wild type and the variant 2. So we have mutation 2. And that brings us to a set of features that we will use. So when we use as an additional input here information such as secondary structure or predicted secondary structure, now we can simply put, okay, this particular residue has a certain secondary structure that's predicted. We can put that into this neural network. We can also ask, well, I have the variant, so I want to predict the effect of a particular variant, right? So how is the prediction changing for that variant, the secondary structure? So rather than putting in the secondary structure for this vector, or the predicted secondary structure, I put in the predicted variant. of the secondary structure. How much, in the central residue here, how much is feature F, in this particular case I discussed secondary structure, going to change upon the sequence change, okay? And that's a new type of input features. Those are the delta features. That's the major novelty here. So there's a bunch of things that Jana uses. She uses alignment information. She uses some biophysical background. She uses probabilities of. She uses secondary structure. She uses solvent accessibility. And this is ultimately shown here. Residue flexibility is another one. And then there are annotation features. Annotation features is this particular residue that has for which I predict an effect, is there an annotation that it's a binding site? Okay? And these are binary features of the type, yes, there's an annotation there, or no. Is this in a region that is known to be an important domain? So-called PolyPhen domain. So this is another, let's call it annotation feature. Again, that's the neural network, and the neural network then has a unit for effect and not effect, and there's a score, and the score ranges. This is scaled simply. We have a unit here that you can scale between 0 and 1. You can scale this between 0 and 1, and you can take all of that and project it onto values between -100, which means neutral, to +100, which means effect. That's historically the way it has been done. And then you ask yourself, oh, well, you do. Actually, I skipped slides. I skipped so many slides that I'm a little bit confused. I'm missing something. So one thing that is specific to the first version of SNAP, so I say that we use the solvent accessibility, but actually the way it was done, we trained two different neural networks, one on residues that are predicted to be accessible, solvent accessible on the surface of the molecule, and one for those predicted to be in the inside. Those were simply two different models. And that turned out to be better because we used different detailed features for these two different models. Clear what I'm saying? So rather than using solvent accessibility as an input, so going back to this image here, so here I only have sequence input, but you can imagine that instead of having 21 plus 1, 22 for the central, you can also for every single one, for instance, add 3 units for secondary structure. You could add, for instance, 2 units for solvent accessibility, buried, exposed, right? And we're not doing the buried exposed part for secondary structure, residue flexibility, and other features. We do exactly add these units. Sorry for the animation. For these two, we do probability of residues triplets. We add a unit. This biophysical features, we add units. But for solvent accessibility, we really have 2 different models because that worked better. For the later version, we dropped that because of simplicity. So sometimes things that work better get computationally more complex and then you drop them. This is an example like that. Anyway, so then how do you see how well they do? Well, there are, in fact, 4 proteins for which a lot of variants had been tested. So I can't remember how many of these added up to 1,000 variants or something like that. So where somebody for these particular 4 proteins had really changed a lot of the residues. And that is how different methods perform. On these dots here mean there was no prediction available. It was only available for this one here. And so essentially what you see is that the SNAP, the machine learning device, is performing at least as good as the others, even better, and that the annotated one here, so that uses these features of annotation, actually improves over this one too. And in detail, you see that, so this is the accuracy versus coverage for effect and for neutral, for buried in red and exposed. So buried, the prediction is more accurate of effect for the model for buried. Again, those are different models. And this one works better, this model here in red, than the one in blue. The green is just in between the two. For the neutral variants, the story is inverted. So now the red works slightly worse, and the blue, so the exposed part, works best. Since we have a difference, so again, in that, say, work prediction for one particular variant, and that's the prediction for another variant, both of those give the same prediction, namely effect, because in both cases, this value here is higher than that value, for case 1 and for case 2. But for case 2, the difference between these two is lower than for case 1. And this difference we can carve into a reliability index. We've done that before. And this reliability index shown here from 0 to simply scale to single-digit integers, so from 0 to 9. 0 means I take, whenever this one is larger here, I call it effect. Whenever it's smaller, I call it not effect. So I make a prediction for every single case. That means reliability of 0. I predict everything. Higher reliability index means I require a higher difference. Requiring a higher difference means for some residues I will not predict, because they don't have that difference. So in this particular case here, that is my prediction, and I will require a difference of at least 0.2, then for this particular case I would say no prediction. Okay? And this is exactly what this graph says here. On this percentage, if I have a prediction for everything, and by the way, this should read effect here in red, for everything, then this is sort of 77% here. If I now look at the 50% most reliable ones, that's the reliability, so the difference being larger than this 4 here, you see that this 50% reaches a level around 87%. Okay? So the whole thing scales. This is for the neutral. The reliability, the strength of the prediction is indicative of how well it performs. And that is what users can take to simply look at the most reliably predicted part, somewhere up here. This is like the hotspot story that I showed you before, where the hotspots were somehow here. In this particular case, there is no biological feature. I'm going to skip the other parts. So this, at some point, Jana worked on this. So in the US, the PhD takes longer. Do you know how long a PhD takes in the US typically?
5 years.
That is a short one. So it depends on the department, but 5 years is not necessarily the average. I would very much doubt that the US average is 5 years. Certainly in biology, much higher than that. Substantially higher than that. The faculty in which I was, the number was closer to 8 at Columbia University. So Jana had worked on this for more than 5 years at this point. And then she sat down and compiled performance. How well does her tool work? And that's what she got. Her tool is here. Those were the tools that were available before she started her PhD, so 5+ years before that point, and that's random. So this is the moment in your life when you get to this point in your PhD where you have to ask yourself, now, is this difference worth 5 years of my life, right? Okay, you also learn, you got a PhD, you learned a lot of things about machine learning, and you created all kinds of things, you created visibility, but my 5 years, and in the US, it's not like, they come at 9 o'clock in the morning and they leave at 5. This is the 24/7, so people work. This is really a heavy 5 years. Well, so one way of looking at it is, this is clearly better than random. And it's also good that after 5 years you're not doing worse than the ones that existed before you started, right? This is already good. But I do admit that this is one of those moments that you hope you will not have such a harsh landing. So we all dream that, here's 100 somewhere. I don't get to 100. Okay, but you want to be somewhere here, right? But here's another way of looking at this story. And the other way of looking at this story is asking, okay, well, SIFT and PolyPhen ultimately use the alignment information. They ultimately use exactly this idea that what is conserved, if you hit the conserved residue, it has an effect. If you're not, they do it in a different way. But let's just, I'm sorry. Let's just look at cases where PolyPhen and SIFT agree. And if PolyPhen and SIFT agree, we call that a simple case. Let's throw those out. Let's just look at the cases where PolyPhen and SIFT do not agree. Before you started your PhD, or Jana started her PhD, people would have had to flip a coin. If the best two methods that existed disagree, which one is true? If they disagree, you know, either you take one of the two, or you flip a coin, right? So let's just look at those cases. And for those cases, you see that now actually those two tools that were available before, in fact, do less well than random. The problem is you cannot pick those out and then throw a coin for those, because you don't know which ones. Well, actually you could, because in this particular case I choose the one where they agree, I disagree. Actually for those, actually you could improve, yes. True, you could improve by throwing a coin for those. And so for SNAP, the performance goes substantially down, but it's substantially better. Right? So that was the moment when clearly it became clear to her that the 5 years were sort of well invested. And these were, when we looked at human predictions, these were many, many cases. Max came, did his PhD, and essentially what he did is he added new data, used new features, much more. He used contact potentials, disordered regions, correlated mutations, low complexity regions, many more amino acid indices, and improved from the light blue to the dark blue, and does overall develop the tool that overall SNAP2 is really much better. And I will leave it at this, and